Us. Happy holidays, happy Salah. Thank you for joining us. So today, what are we talking about? Cancer research for African ancestry. That's something that is interesting. So thank you all for taking time out today, especially today as a holiday. And next, I'm going to introduce our guests. But let's start this conversation. Like I say, together we fight, together we win. Share, follow, and subscribe to Commode Cancer Foundation. So, who's our guest speaker? Good morning.
Good morning.
Oh, Uzwamaka. I'm going to start like this. Don't worry, I'll change it. Don't worry. Good morning, Dr. Uzwamaka Okoli. For the purpose of this interview, you agree I call you Uzwamaka, right?
Yes.
Thank you very much. Good morning. How are you?
I'm fine, thank you. And how are you?
I'm
Great
good
to be
too.
here to discuss cancer research.
Thank you. Thank you for agreeing to join me. So let me give you a brief background on who Dr. Uzwamaka is. Like I said, for the purpose of this interview, we're going to be doing Uzwamaka. She's a research fellow at the University College London, working on the Prostate Cancer UK funded project Radio 3D, led by Dr. Susan Hervey. Dr. Uzwamaka covers the preclinical work packages under this project involving patient-derived cancer models establishment. She's also involved in cancer disparity projects. She collaborates with clinicians to ensure that these models are clinically relevant. Dr. Okoli has a PhD in pharmacology and therapeutics from the College of Medicine, University of Nigeria, where she has developed preclinical tools such as breast cancer patient-derived exographic models. I hope exograft models.
Xenograft.
I always what do you say?
Xenograph.
Xenograph, you see? You can tell I have no business in the science world. She's also a senior lecturer at the coordinator of the Basic and Translational Cancer Research Group, College of Medicine, University of Nigeria. Good morning, Uzwamaka. And honestly, I cannot lie. Medical terms are just too tongue twisting. You guys, you understand what you are saying, but the rest of us, come on.
I'll let you in on the secret too. It's also tongue twisting for some of us as well.
Okay, we agree. But ours is still manageable. So thank you all. Thank you all for joining me today. Let's remain united against cancer. As together we fight, together we win. Let's start the conversation. Uzwamaka, let's start with this now. I'm starting my questions. I hope you're relaxed. Viewers, relax, and let's see what we've got. Now we're looking at more about heritage and cancer. So, Uzwamaka, let's talk about targeted research. Yeah? That should impact the Black Africans, especially us Nigerians. Let's go that far. Can you talk me through global uptake and patient engagement, participation, so patient engagement through participation in cancer-related research? And the reason I've gone after this is I want to understand, and I think viewers would like to understand, how we fare in this area of getting this cancer out of us, if that's how you put it.
Okay, let me talk about the issue at hand so that we can understand the global uptake for how patients participate in cancer research. Despite decades of inequality and under-resourcing in countries, like Africa, we still grapple with substantial challenges. And some of these challenges include high mortality rates, pertaining to cancer in patients of Black Africans when you compare them with the European counterparts. Now, the World Health Organization has projected that annual cancer rate deaths in Black Africans could reach 1 million by 2030. And unfortunately, this mortality rate could be associated with the aggressive nature of cancer pertaining to people of black, African ancestry when you compare with the white European counterpart. Now, paradoxically, despite the equal access to the National Health Service here in the UK, there are more inequalities in cancer incidence and mortality among women of African ancestry when you compare them with other races, especially that of the whites. And more disturbingly, women of, or patients of African ancestry are twice likely to receive a late-stage cancer diagnosis when you compare them to the white European counterpart. So, therefore, and then these late stages of diagnosis often result in less successful treatment outcome. Now, coming to what you asked, shockingly, only 3% to about 14% of patients of African ancestry are represented, in cancer research, more particularly in preclinical studies and clinical trials. And then this disparities is critical, is a critical health equity issue that demands urgent attention and action.
Thank you. 3%. Whoa. It's very interesting because I think I've got an interest in data, and I believe that data informs our discussions, and data informs helping us on the continent get a firm foot in the door because we can justify why. But when we don't have that, how do we argue? Okay, so I'm a bit of advocate from the perspective that knowledge is power.
Yeah.
And that's one of the reasons why the awareness program helps us to educate each other, irrespective of what part of cancer we're talking about. Preclinical studies before trials are important in my opinion. Okay. However, I would like you to explain what they are. So what are preclinical trials and the difference between clinical and preclinical trials? Because if we don't understand it, there's no point us talking about something we're not clear and how it impacts us.
Sure. We talk about this, it's during drug development. Now, before you go into clinical trial, there are preclinical studies. Now, these preclinical studies are testing of drugs outside human. You test these drugs in, modules like cells or animals, while clinical trials are where you test the drug in humans. So before you proceed to clinical trials, you must do preclinical studies to understand what these drugs does to the body and to understand what the body does to the drug, and then also perform some toxicity testing. Toxicity testing, I mean by, to understand what side effects, preempt what side effects these drugs can cause, or, is it safe? How harmful it could be to the body, and then deducing safe, dosaging before you commence, to clinical trials. Now, let me elaborate a little bit on the modules. So, I mentioned you have cells and then animals. Now, cells are derived from patients, or in particular, the cells they use now is called cell line, which is, I would say, in two-dimensional layer. The cancer tissue or the cancer ecosystem is made up of different types of cells. Could be the immune cells, stroma, different types of cells. Now, when you just take one cell and then grow this cell and then keep testing drugs on this, is not sufficiently clinically relevant because it is not capturing all these different talks, these different cells talk, say to each other in the body, thereby promoting, causing it to grow and then proliferate. And then it'll also shock you that these cell lines are derived from, maybe 40 years or something years ago from, of course, mostly predominantly white patients.
Mm-hmm.
And then these cells are immortalized. When I say immortalized, they are somehow modified to, grow and live forever so that you can be testing drugs, which is not so natural to what is in the body. So, and then when it comes to animals, they use, you use mice, canines, that's the dogs, and then, mammals, which, so that you'll be able to deduce, toxicity and safe dosing. However, still, the animal cells still interact with, they're not so, you can't really compare that completely with that of the human. So that is why you can say that the preclinical study aspect is very fundamental because most of the drugs that go to clinical trial fail at 90%. They have 90% failure. So, and the way forward would be to use patient-derived cancer models where you can, collect tissue from patients that generously donated their tissue and then test them, in the lab directly. And then these tissues, they encompass practically all the cell types. You can use this tissue to understand the biology of this cancer pertaining to African, to Black Africans, and so on, to understand how to stop the cancer from spreading. So this is
okay. So I'm going to come back to you, see, because this is interesting because when we had COVID,
yes,
for some strange reason, COVID vaccine came out very, very, very fast. Now, even if you say COVID, even if you want to say the COVID, they had started to do some work before COVID, right? Let's just assume that at least some research had been going on before COVID. The speed in which COVID came out, the vaccine came out, there are today a lot of different conversations as to how much testing was done, how much wasn't done. Okay, fine. But for some strange reason, even the King Charles' grandfather died of cancer. After King Charles' grandfather, so Queen Elizabeth's father had cancer. How many years ago is that? Why are we still having this conversation? It's just, I'm listening to what you're saying, and I'm trying to ask myself, why are we here? If this is a disease that has been going on for such a long time, science has made a lot of progress in trying to change the narrative. However, we're still not getting the Black Afro-Caribbean ethnic minorities in this for these cells and all. Our cells are different. You've got the Caucasian cells, right? You've got the black cells. You've still got the mixed race cells.
Yes.
Every single one of us react to drugs differently. So, sorry, I'm just trying to get that. Why? What's going on? Why are we moving forward? Because if we don't move forward, we're not going to get there.
Well, cancer is very complex. It's a complex disease in the sense that it is heterogeneous. When I say heterogeneous, this patient may present with the same breast. Let me use breast cancer as an example.
Yeah.
Breast cancer and then breast cancer. And then this patient will also come with the same breast cancer. And then you use this drug to treat this person. This person may, show complete response to the drug. Well, this person may be, resistant to the drug, will not. Now, there are things that drive cancer in us, in the cells of human, and it's called the genes.
Mm-hmm.
This is one of the cancer there are several cancer hallmarks, about 14 of them right now. But one of them would be the gene alteration in the cancer environment, which drives, this certain cancer. So chemotherapy, as we all know, targets it also targets both normal and cancer diseased tissue. And like I said, African ancestry, they present mostly at late diagnosis.
Mm-hmm.
So endeavors are being made to, try to target these genes directly. Is that, to target the disease directly, but it's actually it's a complex situation.
So can I ask you, when you said genes, are we talking about this conversation or this work going on genomics? Is that where it comes from? Is that part of it?
Okay. Genomics means the study of genes.
Okay, so sorry, I'm finished.
Go on, sorry.
Okay, no, so if it's genomics, that means that there's a conversation, because I do know that there's a lot of conversations going on genomics at present, and its impact and its benefits long term. So there is a need now for us I'm just asking of us and the African origin,
yes,
to really get ourselves in this conversation if we're going to be able to if we're going to ensure that our genes are represented in the decision, in the formulas that are going to go forward. Does that make sense?
Yes, it does.
Okay.
Because there's a lack of genomic data out there for pertaining to African ancestry or African origin or Black Africans. And it's through these modules, doing genomic studies or molecular studies, that there's something called central dogma of life, where in the human cell, there's the DNA, which I call the genes. And then this DNA translates to mRNA. mRNA now translates to proteins. Now, these proteins could be in the form of, let me use a layman's, can be in the form of insulin and every other proteins that function in the body. So, particularly maybe this gene DNA translates to genomic studies, and then the mRNA translates to transcriptomic studies. This enables us to understand how patients respond, we researchers respond to drugs, and how the cells signal each other, and how we can target that particular aspect of the gene with the drugs to understand. But then when you lack data, of this, of some sort, how do you begin to understand the, biology underlying that of African ancestry. So,
You know why I laugh? Because the next thing I was going to say is that I think everything about us as Africans, we need to sort out our data. Because
yes, we do.
until we sort out our data, we're not actually giving ourselves. So it's nobody's fault. It's us. If we don't sort out our data, make sure we have accurate data, make sure we are sharing reliable data on all aspects, in all aspects of not just cancer, but everything, we're not going to be able to resolve our issues as timely as developed countries are doing. Is that fair to say that? Okay.
Yes, it is.
So
Because
what are patient-derived cancer modes then? Is that what you were just talking about?
yes, yes.
Okay.
And there are different types of patient-derived models. There's patient-derived models are where you access tissue from patients,
Okay.
and then you use this tissue in the lab, to do drug testing. And there are different aspects of it. You can crush the tissue and harness cells, which is monolayer, but then it will make you understand the different roles of certain things you are interrogating. Whereas there are explants where you have the tissue in the hole, and then you culture them in the lab and test these drugs. And this is also good because it has all the cells in there, in it. And there's also where you can actually take that tissue and then grow it into mice or into animals, and then study the human tissue in the animals. These are called PDXs, that's patient-derived xenografts. The other ones I mentioned were explants and also spheroids or organoids.
Okay, so, let me not get myself confused. I think a lot of us viewers, we're not all medical people. We're normally just ordinary people trying to understand how to navigate, or at least know. See, the point is making sure we can ask the right questions. It's no point going before a doctor or trying to discuss all these things without understanding this impact on us. Okay, so I'm going to ask you my last question. And if this question goes, and it's a standard question I always have to ask, is share in one sentence for each of the three aspects of this a take home for government on policy or generally, a take home for cancer advocacy in Nigeria specifically, and a take home for patients, family, and friends. I always like to have one thing that we can actually say, this is what I've learned, and I think I can work with this. I can use this to help people. Because there's no point talking about advocacy if we're not giving out the tools that are important to navigate going forward. Thank you.
Okay. I'll say for policy, that the government also review, like they have policies for clinical trials. They should also be inclusive of preclinical studies pertaining to, African ancestry. I'm not just talking about Nigeria, I'm talking also globally. Then, create infrastructures where that is sustainable. For instance, biobanks, where biobanks would be, where you collect such data. Patients generously donate, tissues, samples, clinical history, data that will be securely, kept and stored, and then the government should provide fair, accessibility to this infrastructure where we can collaborate, within ourselves, with approved researchers, and also internationally, globally. And
One
of course,
sentence for the next two, please. One sentence.
one
One
sentence,
sentence.
and then each research should involve patients, the patient engagement, to understand how important it is for us, for them to be also part of the study, from the onset to the end of the study as well. And what was the last one?
That's fine. That's fine. I want to say a very big thank you to you. Great works are performed not by strength, but by perseverance. I want to say thank you very much, Dr. Uzuamaka Okoli, for joining me this morning and educating us on these cells.
Thank you very much. Thank you for having me.
You are most welcome. Follow us on our social media handles as we share various activities happening across the country, on Common Cancer Foundation and all the NGOs across the nation. Together we fight, together we win. We want to say to you, happy Eid. It's Salah, and I hope that everybody has a fantastic day. Nigeria is on public holiday, so you can follow us on our foundations, share, subscribe, and click on the notification buttons on all our videos. This will be out in the next few days. And you can see us on our Instagram, Facebook, YouTube, and LinkedIn. Let's have a chat. There's a live chat out there. We can all get together and learn. Help us educate family and friends this 2024. Thank you all for joining us. Have a fantastic day. See you in 2 weeks.
Thank you.
Thank you. Our message is very clear. Together we'll fight. Together we win.